Hello, my name is Billy Dunn. Today is May 28th, 2018. I will be discussing the lab exercise for bacterial morphology and staining techniques. The first technique I performed was the direct and negative stain, which requires only one color stain. I used these two stains here, or two slides. One slide was used with violet color, and the second slide was used with a Congo red dye. Two different techniques, but very simple, very quick. Before I put the dye on, I swabbed my inside of my mouth, or actually my teeth, to get the bacteria. Put the swab on the, if you can see the little circle. I used the two dye techniques. One requires a little bit of heat, a little tea light, in order to kill the bacteria and make it stain or stay on the slide. Once I did that and rinsed off the dye with a little bit of water and a pipette into a little dish here, I let it dry, and then I observed on my microscope what bacteria I had in my mouth, and that demonstrated coccus, the small little beads, and the other slide showed bacillus-shaped bacteria. The second gram staining technique I did was the differential stains, and that I used 3 different slides. I used my my broths, my E. coli or Escherichia coli, Staphylococcus cerevisiae, and Staphylococcus epidermidis. And what I did was I used my loupes and I dipped it into my broths. But before I did that, I always want to sterilize the tip. So what I did was sterilize the tip with my flame, dip my wand carefully inside. I'm not going to reuse this. And then in the little circle, I streaked it in a circle. Before I put my cap back on, I went over the fire once more over the lid, returned the cap, and with the differential stains, it's a different type of technique. You have to use 4 different color stains, and first you use the gram stain number 1, which is crystal violet. You put a drop onto the circle, you let it sit for about 60 seconds, and then rinse it off with distilled water. Then you do the same with PVP iodine, and then the decolorizer, and then last is the safranin. So once you do that, each time you're letting the dye sit for about either 30 seconds or 60 seconds, then you're rinsing it off with distilled water, and then you let it dry, apply the next dye, let it dry completely, and then that's when I proceeded to look under my microscope to see what's growing on my plates with the differential stains. Unfortunately, that was not a successful gram stain for me. I'm not sure if I washed the all the bacteria off in the process of staining with the 3 or 4 different colors, but it's a trial and error. All you can see is very small coccus. I would say it's very like pin dots size on the slides at all magnifications. So the quicker direct and negative stain proved to be more successful and showed bacterial growth instantly. Again, my name is Billy Dunn. Today is May 28th, 2018, and I have discussed and demonstrated quickly the bacterial morphology and staining techniques. Thank you.